thanks everybody for coming, whether in person or online, to our Twist event here. Just a few housekeeping items. All lines will be muted. If you do have a question, please put it in the Q&A box, not the chat. So we'll answer those after each of our talks. Following this event, there will be a short 1-minute survey. If you can please fill that out, it'd be great, very helpful for us to know how we're doing. So yeah, thank you. I'm Tim Barinholtz. I'm an NGS field application scientist here at Twist. I'm going to talk a little bit of background about what Twist is doing to enable advances in epigenetic analysis. So Twist is a relatively new company to the NGS space. We were founded in 2013. We IPO'd in 2018. We have offices all around the world. We are headquartered in South San Francisco. Our main technology is that we are able to print high-quality oligos on a silicone chip. In the typical space of a 96-well plate, where typical oligosynthesis happened, we now have a silicone-based platform that can print 1 million unique oligos. These oligos have a industry-leading 1 in 2,000 base pair error rate, so very, very high quality. And we use those to tackle challenges in a variety of spaces. So today we're going to talk mainly about NGS, but we also can provide gene fragments, clonal libraries, oligo pools, and we have a biopharma and data storage department. Our goal for NGS is to provide the maximum efficiency for all sequencing runs. So we are specialized in hybrid capture arrays, where we are providing a series of targets, and we want to sequence them to an even depth, which we call uniformity. So if you take a typical capture target with 3 targets where you're trying to get a 30x depth for each of your targets, you want to minimize over-sequencing or extra data that you do not need, or under-sequencing where you're going to throw away the reads that were dedicated to that target because you can't call your SNP or methylation call. And obviously, off-target is never appreciated, because that's all wasted data. So specifically in epigenetic analysis, Twist has come up with an end-to-end workflow for methylation sequencing. So we have an end-to-end workflow. So we started by partnering with NEB, leveraging their expertise in enzymology. They have a fantastic product, their EMSeq. So we partnered with them to develop a library prep that can provide enzymatic conversion, followed by Twist's total purification beads. You then move into a target enrichment process with Twist with a fast hybridization system, our new methylation enhancer, followed by a custom panel, or what I'll talk about a little bit later, our new methylome panel. We wanted to partner with NEB specifically because of the advantages that enzymatic conversion provides to the gold standard, which is bisulfite sequencing. So the gold standard bisulfite sequencing, you see compared to enzymatic, you get roughly the same conversion rate. So we can say we have a little bit higher conversion with enzymatic conversion, but the main goal here was to show that you are still converting at the level of what the gold standard is bisulfite sequencing. The real advantage comes from improving the degradation of your DNA. So typical bisulfite sequencing highly degrades your DNA, so you're unable to do low input samples, such as cfDNA or FFP or highly degraded samples. So when we were looking to develop a methylation system, a methylation sequencing system, we started looking at bisulfite controls where you're getting 8.8 nanograms per microliter out of your library prep compared to the NEB EMSeq, where you're getting 112 nanograms. So you're retaining a lot more of that DNA, the precious DNA that's going in your system, and it allows a lot less input. Additionally, we don't see the GC dropout that you typically get with bisulfite sequencing. So if you look across the GC spectrum between enzymatic and bisulfite conversion, you really retain this high GC content that you lose once you convert the unmethylated Cs in your samples. And if you look at the comparison between a hypomethylated or hypomethylated DNA, gDNA targets, you can see that the enzymatic conversion retains the high coverage in those high GC areas. Now, getting more specific to sort of hybrid capture statistics, if you look at how many of your targets are actually hitting your depth, so say 30x coverage, that is higher. You get the more uniform coverage, the full dating base penalty, so you're getting all of your targets are hitting the same depth of coverage across all your entire panel, and you're not getting peaks and valleys between your different targets. You're getting less off-target effects, and you're having fewer duplications. So this is all saying that you're more efficient in sequencing, and you're able to put more samples onto the sequencer and save money ultimately in your sequencing costs. So we had a decision to make whether we wanted to do pre or post capture conversion. So I'll start with the bottom where you have post capture conversion. So this relies on first performing library prep, followed immediately by your target enrichment or your pull down of your targets, then followed by your conversion of choice, then finally with the PCR amplification. This is different to a pre-capture conversion where you, following library prep, you immediately convert all of the unmethylated Cs to Ts, followed by the amplification, then your target enrichment. The benefit of doing a pre-capture conversion is it allows for even lower input into the system because we're able to do this amplification post-conversion, and then we rely on a more complex panel design that includes 8 individual oligos that pull down your targets, and this allows for greater library complexity and lower input. So just to go over our design process for our methylation panels, we start by taking each target, each CpG site, and considering it as unmethylated, fully unmethylated or fully methylated as a state. Then we go through in silico the conversion and the resulting PCR of that product and what it will look like. And then in that space is where we design our probes. So in your panel for your target, for your CpG site, you actually get 8 pieces of DNA that target that site, whether it's fully unmethylated or fully methylated. And that allows us to grab all of the states in between, whether it's 50-50 methylated, 25% methylated, and really and look at DMRs specifically. This is an example of what that data would look like. This is a contrived experiment where we took hypomethylated cells and hypermethylated cells, or 0 and 100% methylated. We mixed it in different proportions to give us 25, 50, and 75% methylation. On the left, you see the general capture statistics. So again, how many of the targets get 30x coverage, how uniform each of the captures were, the off-target, and the duplication rate. So pretty flat across the methylation, how methylated the samples were. And if you look at the actual call pileups in a program like IGB, you can see that it retains that same profile. So the 100% methylated sites appear to be 100%. So qualitatively, it looks like it's retaining those differential methylation states of the contrived samples. So I really want to talk about our new product, the human methylome. This is a very exciting product that we just released, and it is a very large panel. It's an exploratory research panel. It's 123 megabases. It's a very, very large panel. It covers just under 4 million CpG sites. With shadow coverage routinely for our alpha customers, we're hitting about 6.5 million CpG sites, so just a massive amount of CpG sites. It's very up to date. It's recent, very recent panel. So we scrubbed, UCSC Ensemble ENCODE, worked with key opinion leaders. A lot of people ask us what is in this panel. The easiest way is just to ask us for the annotated bed file, and you can scroll through and see if your targets of interest are in there and determine if this is a panel for you. On the right, you'll see just kind of where the targets lie, whether they're in CpG islands, shores, shelves, or open seas. Since it's such a big panel, we also get a lot of questions about, well, how much sequencing do I need to throw at a sample to get data out of this? So here's kind of a way to think about it. So on the bottom, you have the raw coverage that would come off the sequencer. So how much sequencing space you add to it. So the lightest gray bar will be the coverage that as a researcher you care about. That is how much coverage you will have on your target at the end of the day after you go through alignment and deduplication and all that. So at 150x raw coverage, you're getting about 57, so about 50x coverage on your target. So that's what we would kind of suggest to most researchers for their methylation calls. And then at the end of the day, how much does this actually cost in sequencing space? So this is not including the panel of the workload, but your sequencing center, for example. And this is using the cost on Illumina's website for the various flow cells on the NovaSeq. At 150x coverage, you're able to fit 130 blood samples on an S4 flow cell, which comes out to about $110 per sample. Sorry, yeah, $110 per sample. And then with the entire package, it's very, very competitive with what we're aiming at eliminating from the market, which is the Epic 850K array. So this is how it kind of stacks up to the 850K. So we overlap with just under 95% of the CpGs targeted in the 850, but we of course provide another 3.1 million, and with shadow coverage, it's closer to 6.1 million additional CpG sites. And if we look at the correlation between the two, next one, we can look at how the human methylome from Twist correlates with the 850. We have a R-squared of 0.973, so correlates very, very well. If you cut down on the amount of data points, you can kind of see things. You'll see they really diverge at the 0 and 100% methylation calls. And if we look at that in a slightly different way, easier way to look at it, next, you get this higher dynamic range in the human methylome. So the array is very bad at calling these 100% methylation calls with 0% methylation calls. The noise in the system, you get saturated towards the 100% methylation, and it's too dim at the 0% methylation. So the NGS provides you that ability to truly call hypomethylated and hypermethylated states of your targets. This is a really quick example to go over. This is in the BRCA1 promoter region. In the green in the middle, that is the Twist human methylome, and on the bottom you have the 850K. And most of the calls from the human methylome, you're in the 1%, 0% region, and the Epic you're seeing 2, 4, 5. And then here in this red circle, you have 0% by the human methylome and 18.5% from the 850K. And if we drill down into what this is actually, is this a true 0% call, or is it a zero coverage target for the human methylome? When we look at this one site, we have 145 individual calls for this one site, and we're calling it 0% methylated. And what it turned out to be is that in this site there was a C-to-T mutation in the BRCA1 promoter region that we can call because we are actually capturing the plus and minus strands, so we're able to call SNPs as long as they're at a high enough frequency, which you can't detect in an Epic array because you can't call SNPs. So any C-to-T mutation is called as an unmethylated state. So you get a lot more dynamic range. You're able to you can call SNPs, and it's just on higher performance, and you get way more methylation sites. Luckily, in your region, you have a fantastic team to support you here at Twist. Sarah Sodden is your NGS sales specialist, who is supported by Alan Fox, which is your local FAS, and then Brendan Hicks, who can help you with all your SynBio needs. And now I'd like to introduce our speakers for today, what everyone's been waiting for. First up is going to be Dr. Michael Corley. He's an assistant professor at Weill Cornell Medical College. His lab focuses on host-epigenetic mechanisms in immune cells reprogrammed by HIV infection, and how unique cell type epigenetic signatures impact innate and adaptive immune cell responses and longitudinal brain health outcomes following HIV exposure. And then followed by Dr. Jeremy Rock, who is here with us in person. Dr. Jeremy Rock is an assistant professor at Rockefeller University and heads the Laboratory of Host-Pathogen Biology. Got his PhD at MIT, did a postdoc at Harvard, and his lab is now laying the foundation for new therapeutic strategies by investigating how Mycobacterium tuberculosis causes tuberculosis and evades current antibiotics. So please help me welcome Michael Corley.
Thank you for the invitation. Let me see if I can get my slide up. So today, I wanted to give a brief overview kind of our lab's application of looking at DNA methylation states in the context of both HIV infection of the host, and also an application that we've recently adopted and tried with the EMC Twist protocol to look at potentially pro-virus DNA methylation states. So for today, briefly, I'm going to introduce first a project that we just finished up. And it's a unique project that we basically applied an epigenome-wide scan of participants that we had recruited in an international study looking at acute HIV infection. So these are participants that are essentially identified at the earliest stage of HIV infection. And in that study, we kind of began to ask some questions regarding how soon after infection, potentially, that host epigenetic changes to DNA methylation states may occur in the host. And another unique aspect of that study was we also were able to address the question of whether early treatment, so in the context of HIV infection, currently antiretroviral therapy is very effective at suppressing the virus and letting people live a long life comparable to before the advent of antiretroviral therapy. So we were able to ask the question whether early initiation of therapy in these individuals seemed to reset or mitigate some of the host epigenetic changes that may occur, since we had longitudinal studies from the study participant. And then in the second part, I'll bring up a newer part where we're starting to look at potentially addressing some of the challenges of actually looking at integrated provirus. So HIV being a retrovirus, it integrates into the host genome, and particularly looking at CGs that may be methylated of the provirus and potentially how that relates to different viral states. So just a brief background for those that aren't aware, currently global HIV and the AIDS epidemic is not over. It continually, I think, continues to evade a vaccine. So currently there is no effective vaccine for HIV. Currently as well, those individuals living with HIV continue to harbor the virus despite having effective antiretroviral therapy that prevents the virus from being transmitted. These individuals have to daily take medication essentially to suppress the virus and keep in a state that is keeping the virus essentially latent. And if they stop taking medication, the virus rebounds and comes back. In terms of our interest in the lab, we've been quite interested in asking two questions. First, potentially how HIV disrupts the host epigenetic landscape, in particular cell types. We think that this question, I think is quite fundamental to achieving potentially new therapies or even cure strategies to overcome the virus. And second to that, we're also, I think, looking at longitudinal outcomes. So a lot of individuals living with HIV currently are experiencing quite a few HIV-associated comorbidities. And we think that a lot of these potential changes may occur during the early establishment of viral infection. So essentially, the host being quite important at the early stage, determining longitudinal outcomes and how they respond to the virus. So for the first part, this is a study we just completed, and we really asked, I think, a unique question in terms of potentially looking at how soon after HIV infection, HIV-associated epigenetic changes may occur in blood, utilizing cell sorting. So we had a unique cohort, which was in Thailand here that I'm presenting. This unique cohort essentially relies on the fact that they're able to screen individuals in real time in Bangkok, Thailand, and utilizing a different type of screening protocol, they're able to identify people at the earliest stage of acute infection. So for the study here that I'm presenting, we were able to look at 22 individuals that had a median day since estimated exposure of HIV of 17 days. The earliest captured data profiled in the study was a participant that had been identified at the earliest stage of HIV infection, which is referred to as FUBIG1. And this was estimated to be 9 days since exposure. So this individual was HIV RNA positive, but had yet not mounted an antigen response. The cohort is also, I think, uniquely positioned to immediately initiate ART treatment in these individuals. So they go on therapy quite early. So essentially, the day that they come into the study and enroll, they're given antiretroviral medication to suppress the virus, and they're longitudinally followed on subsequent study visits, and biospecimens are collected. For this study, the most shocking part, I think, when we looked at the data was that the acute HIV-related DNA methylation changes seemed to occur early, but comparable to, or contrary to what we initially thought, the majority of changes occurred in monocyte cells that we had self-sorted from these individuals compared to Cd4 lymphocytes. Interesting that the Cd4 lymphocytes are the cells that are infected by the HIV virus and integrate into the genome. It seemed like the innate immune response harbored the majority of DNA methylation differences that appeared. The Venn diagram I'm showing you there is kind of the overlap. So there were changes essentially that were shared between the two cell types, but the majority of those changes seem to be harboring in the monocyte cells comparable to the CD4 T cells. And then subsequently, we started to ask questions related to potentially persistent changes. I'm providing an example here of one of the lasting changes that we identified. And this change that we observed was at a regulatory region of the EZH2 gene, which is essentially an enzyme that participates in histomethylation and transcriptional repression. And intriguingly, in the literature, there's been a lot of work on EZH2 looking at reactivation of HIV from latency. And I'm showing you here on the bar plot just an example of one of the CpG sites among the regulatory region of the EZH2 gene in HIV uninfected individuals in green here. And then these are the individuals that were at acute infection staged at the different feedback stages. You can see there seems to be a significant hypomethylation in those individuals during acute infection. And then when those individuals are placed onto ART treatment and the virus is suppressed, the methylation state of this particular CpG site in the host seems to maintain at this lower level comparable to the HIV negative individuals. And then subsequently, we had a unique cohort as a comparator from pre-existing data. This is in the context of chronic infection. So this is the majority of individuals living with HIV are chronically infected and then placed on therapy. So this is just showing you a comparison of those individuals pre-antiretroviral treatment, chronic infection, and post-antiretroviral treatment. As you can see as well, this particular gene seems to be hypomethylated in association with HIV exposure. And then just as a comparison here, we had a unique opportunity to harbor DNA samples from one of the individuals that has been cured of HIV through an unconventional stem cell transplant therapy on the Berlin patient here. Just showing you kind of a reference point of the demethylation state in this individual. But I think the take-home message is essentially that potentially lasting changes to the host epigenome may be placed down into the host, and HIV may be utilizing these potentially, I think, to either maintain evasion from the immune system or to essentially repress itself and hide. And then, as I said, the one thing that we're starting now, I think, to really get interested in terms of methylation is methylation seems to, at least of the host, be informing us on clinically relevant outcome measures that I think are quite interesting. So just by taking the methylation from the study that I introduced, I'm showing you here examples of relationships between, I think, clinical variables that are quite important, I think, for outcome measures at the set point of viral infection in the context of HIV. I'm showing you here that essentially by taking the methylation state of interferon-related genes, we were able to see significant relationships between the methylation of monocyte cells and the participant's viral load, suggesting potentially that mounting of the antiviral response by the innate immune system associates with viral sensing. So, essentially the demethylation of these sites and the transcriptional upregulation and activation of interferon pathways seems to be associated with methylation. And just by taking the methylation, we're able to essentially infer the different viral load that the participants had during acute infection. The other interesting aspect as well, and I think one that is quite important in the context of HIV, is Cd4 count. So Cd4 cells are infected with HIV, and they predominantly are eliminated from the immune system. And for individuals, you want to maintain a high Cd4 count to have better outcomes. So by looking at the innate immune response methylation state, we're able to see significant relationships with patient Cd4 count at particular interferon regulatory genes. And then we also had clinical data looking at a CD4-CD8 ratio, which is another important clinical variable in relationship to HIV. And we similarly saw, looking particularly, again, at interferon-related genes, significant relationships between the methylation state and the CD4-CD8 ratio, suggesting, essentially that methylation seems to be a good proxy of general well-being and health of the immune system in the context of HIV. And then just briefly taking you, I think, to the second part, what about the HIV Pro-Virus? So as I said, the majority of individuals living with HIV have basically HIV integrated into their CD4 cell DNA. There's been a lot of work, I think, initially early on looking at CpG methylation in the context of parasitic sequences in terms of endogenous retroviruses, showing you that silencing of those endogenous retroviruses is mediated by DNA methylation. In terms of HIV, there's been in vitro work showing that CpG methylation can silence proviral expression in latency models. When we start to look in vivo, the story becomes a little more controversial, suggesting some studies showing that DNA methylation doesn't seem to be playing a role in viral suppression or reactivation, and then other studies, I think, are contrary to that. But I think a lot of these initial studies suffered from the fact that they relied on traditional PCR-based methylation assays that needed a lot of DNA material and bisulfite conversion protocols that essentially degraded a lot of the DNA. So the signals were, I think, challenging to overcome. So for us, we started to ask the question whether the Twist EMC protocol could potentially address some of these challenges. And what we did was basically apply the Twist targeted enzymatic methylation sequencing protocol to HIV. And we asked some pretty, I think, fundamental questions. To start, can we enrich for enzymatically converted HIV viral sequences? And then could we essentially extend coverage of CpGs across the complete provirus? So the HIV virus itself, if it's intact, is around 9 kb. So essentially a lot of the studies that have been conducted have predominantly looked at the LTR region and focusing on a couple CpGs. But since we have the ability, to enrich for multiple sites, we want to see if we can get better coverage. So what we did is we first started with a cell line based approach. So we utilized a JLAT T-cell clonal cell line that had an integrated copy, one copy, so we know exactly the sequence and where it is. It's replication incompetent. Here I'm just showing you that the unique thing with these cells is they're GFP tags. So if you reactivate them, they light up green. And then we also had another cell line, an ACH2 cell line, which is an older HIV model of chronic infection. It's replication competent and has multiple integration sites. So we took DNA from these cell lines, designed probes, and then asked the question whether we could look at methylation of the provirus. Here I'm showing you data of our EMC targeted experiment of converted HIV provirus that's been enriched utilizing the targeted Twist protocol. In the IGV plot here, for those that aren't familiar, I'm just showing you the top panel is basically the whole length of the 9 kb or so HIV virus from the JLAT cell line. Here I'm just showing you the coverage, the histogram of the reads that were mapped. We obtained utilizing the sequencing approach, very cost effective, over 250x coverage. And in green and red here, I'm just showing you the location of the CpGs. Another way to look at that here is when you convert and analyze the data, the blue plots are essentially each CpG, and the height of the histogram plots here are essentially the level of methylation. So I'm showing you here two conditions where we had a control condition and then a reactivated condition. In this pilot experiment, the caveat, I think, to the reactivation experiment, you would expect, right, if we reactivated and the virus come back, you'd expect more dramatic differences in methylation. Just for this experiment and just for time, we weren't able to sort the GFP positive cells. So, this reactivated condition would include a pool of DNA from both virally transcribing cells and cells that potentially didn't get reactivated yet. But in general, you can start to see some drop in methylation here at the LTR, which has been previously been described to be linked to reactivation of HIV provirus. But I think the take-home message is that the method can be used to study the epigenetic regulation of HIV latency by looking at cytosine methylation patterns. And as you can see here, we get quite good coverage across all the CpGs of this particular viral sequence of this cell line. When we applied it to the ACH2 model, we ran a control condition. So on the top plot here, I'm showing you a methylated control condition of ACH2 HIV DNA. We utilized an enzyme, CpG methyltransferase, to artificially methylate all the sites. So you would expect, right, that methylation should be greater than 99%. And we do see that when we artificially methylate, suggesting that, again, the method is quite good at picking up a quantitative assessment of methylation. And then two conditions here for the ACH2 cell line. The interesting thing with this cell line is there seems to be not much methylation deposited at the 5' region of the virus. And more importantly, too, the cell line seems to be spuriously transcribing, so it's not a latent cell line. So it is making HIV RNA quite regularly, suggesting that even when you stim, the cells themselves are already making HIV. So, you wouldn't expect methylation to be high, essentially potentially at a shutoff region of the virus. But just showing you here again, too, the consistency as well in the two conditions, in terms of independent replicates being quite, I think, accurate and detecting the same level of methylation across CpGs of the provirus. And then to end the talk, this is something we're working on now, is potentially how would we apply this in the context of patient samples. For HIV, this is quite challenging because there's a rarity question. So essentially 1 in every million or so cells is infected with provirus. And then also another further challenge is, as we know, I think looking at viral evolution in the COVID-19 pandemic, HIV being even more complex, the viral sequence diversity even makes this more challenging in terms of the mapping. So, it's really having access to samples that have already sequenced autologous sequences and being able to map to those and utilizing those. But I think as a first step, we just asked the question in terms of the rarity question, so whether we could capture HIV provirus DNA methylation, since we are using capture probes, in a dilution kind of experiment. So we took approximately 2 cells with Hiv Pro-Virus, and they have one integrated copy, and we diluted this among a pool of jerk cat cell DNA that doesn't have HIV virus, and then just asked the question with the pull-down capture approach, could we even enrich these sequences? I'm showing you here our first experiment where we attempted this. As you can see here, the coverage isn't as great, and I assume some of the DNA material through the sonication is lost through the protocol, but it does seem to suggest that if we do have, I think, greater than 1 copy of HIV DNA or even higher, we will be able to, I think, apply this in terms of being able to enrich rare HIV Pro-Virus from patient samples in a DNA pool. So just to summarize, the host cell type specific DNA methylation patterns seem to be impacted quite early during HIV infection. And this is, one angle from the host that we're quite interested, I think, in also applying a twist new protocol looking at the full human methylome to our cells from patients living with HIV. And then the second point, despite effective treatment with ART, there's seem to be persistent immune cell epigenetic dysregulation associated with HIV that seems to persist, even though these individuals are living in a suppressive state and not harboring HIV RNA transcription. And this is another thing too that we're quite interested in figuring out, potentially these long-lasting, durable epigenetic perturbations of where they are and how we may be able to modify them to, I think, be able to boost up the immune system to get it back to a state prior to immune suppression. And then I showed you, I think, our initial work on establishing targeted EMC to establish DNA methylation of the Pro-Virus, looking at the viral side of things, and potentially how this could be applied to look at Pro-Virus in human clinical biospecimens. And here I just ended with a paper from a group out of Harvard, that's essentially looking at a unique patient population of individuals living with HIV that are able to spontaneously control HIV without having to be given therapy. And in the paper, it was interesting that, that they're also highlighting here, there's the possibility that HIV promoter methylation, which has previously been shown to induce proviral HIV silencing in vitro, may contribute to durable transcriptional repression of proviral sequences from these people that are referred to as elite controllers. So, if we can start to, I think, get a better understanding of the landscape of methylation in different settings, I think we can start to harbor this to potentially durably suppress HIV. And that, just to end the acknowledgements, here's my funding sources, our lab at Weill Cornell, and then for the international work, a large team of individuals that participated in RB254, and particularly the sub-study participants for providing biospecimens. Thank you.
Just as a reminder, if you have any questions, please put them in the Q&A box. So we'll take any questions for Michael. I have one. Since endogenous retroviruses tend to be silenced by DNA methylation, is this a possible avenue of therapy with HIV, is to force it into a hypermethylated state?
Yeah, so I think currently that's, some of the interest that some of the groups are working on, and potentially utilizing the CRISPR-Cas9 system of silencing to target potentially DNMT to patient endogenous retroviruses, and to get them to essentially durably suppress. Another approach, right, is potentially, could you cut out the virus? I think that's a complementary approach, but one of the other options, right, is to just try to induce it into a similar endogenous retrovirus state of permanently being silenced.
And do you have any numbers as far as how many more CpG sites you were able to actually target when you did the EMC protocol from Twist compared to typical bisulfite?
Yeah, so for us, the unique thing, right, is that it's kind of giving us information across the whole region, whole Pro-Virus, any CpGs that are basically in that 9 kb sequence. Previously, we were utilizing, bisulfite PCR methods, which at most, you capture around 5 or so CpGs when you do that, and then you have to call any PCR and clone. With this method I showed you, it looks like we capture all of them. So, just comparatively, there's 50 CpGs potentially in a viral sequence, and previously we were getting 5. It's, I think, quite a lot more data that we're able to look at, and, essentially look at methylation sites that we didn't think maybe that there was action at. The method would kind of give us, I think, insight to that. And then having the sequence too, I think is an added benefit, like you brought up, to look potentially at sequence diversity.
Okay, help me thank Michael again for his excellent talk.
Thank you.
Okay, our next speaker is Dr. Jeremy Rock. They'll be talking about genome-wide titration of essential gene expression reveals diverse vulnerabilities of mycobacterium tuberculosis.
So thanks, Brennan, for the invitation to come speak. We've got a lot of libraries with Twist. It's a fantastic product. So we're excited to kind of show you what we've been doing with it. All right. So we can go to the first slide. Okay. Yeah, that'll work. So my name is Jeremy Rock. I'm an assistant professor at Rockefeller University, and I study tuberculosis. So I'm in a room with a bunch of DNA synthesizers. I have no idea who's on Zoom. So what is TB? So I'll just call it TB, or tuberculosis for short. It's caused by a bacterium called Mycobacterium tuberculosis. It's primarily a pulmonary disease, or a disease of the lung, but it can infect effectively any organ in you to which it can disseminate. We are the petri plates for this bacterium. It's a very ancient disease. The Greeks called it physis, consumption, the white plague, Pott's disease. So it's been with humanity for a very long period of time. And what's interesting, it is an obligate human pathogen. The only place it lives is inside of us. So theoretically, this is an eradicatable disease. Next slide. But we're nowhere near that. We have not eradicated TB. TB has remained a global pandemic. These are the numbers from the WHO for 2020. Next slide. Don't need this. So in 2020, there were about 10 million new active cases of disease every year, about 1.5 million deaths, and about 500,000 of these cases were multi-drug resistant. Unfortunately, since COVID hit, these numbers have actually, for the first time in decades, actually gone up, unfortunately. Next slide. Okay, so TB is a bacterium, but this is not E. coli, right? This is not a model organism. It is an organism. And in fact, here's a phylogenetic tree of life. And what you can see by mapping the distance between these lines is that TB is more different from our model bacterium E. coli than humans are from plants. Nobody would argue that we're going to study plants to understand humans. Same way to say we can't just study E. coli to understand TB. So that's why we study the actual organism. And to be fair, it is a very difficult organism to work with. It's 24-hour doubling time, more like tissue culture cells, and you'd have to work in a biosafety level 3 environment. Next slide, which I think is super fun. This is what it looks like. So these are a few of my lab members in the BSL-3, and you can see they've got N95s or respirators on. So again, we are the petri plates on which this bacterium grows, and that's why we work with it in a BSL-3 lab so that we don't infect New York. It's very safe, I promise. Okay, so next slide. What are we doing with the libraries that we get from Twist? So we're geneticists and biologists, and our motivation is that we're going to discover new biology that facilitates the pathogenesis of this bug. And then that's going to allow us to discover new drugs that perturb that biology, and we're going to try to help cure this disease. And the problem is, this sounds great. It's a great way to get grants. It's a great way to motivate your work. But next slide. To a first approximation, it doesn't work. For bacteria, we can't really start, or we have not yet been successful in a clinically meaningful sense of starting with a target and ending up with a drug. This requires what's called target-based drug discovery to work. And as a biologist, this is usually how I motivate my work. And if that doesn't work, then this rings a little hollow. So why doesn't this work, and can we get it to work? So next slide. Okay, so why doesn't target-based drug discovery work? Why can't we start with a target and end with a drug? So what you do is you find a target that, say, is essential for the bacterium, you purify it, you do high throughput in vitro screening, identify small molecule inhibitors of that enzyme, and then dump these inhibitors on the cells, and then nothing usually happens. So why does nothing happen? Well, the compound could not be able to get into the bacterium. The bacterium have this outer envelope that's highly impermeable. The compound could get in and it could get degraded, or it could get in and then get pumped right back out, right? So these are all real. These are real reasons why this method doesn't work, but they're all sort of blame the compound approaches, right? If only I could find a better compound, I could get this method to work. But we started to think about, well, we're missing the other side of the coin here. Next slide. What about the targets themselves? Okay, so let's say in this fictional scenario, we have two targets. Both of them are essential for the bacterium. So if we knock them out, the bacterium would die. And we had an inhibitor that could inhibit both of these things at sort of 2/3 level inhibition. And if we inhibit the gray target, the bacteria live. If we inhibit the black target, the bacteria die. So what we would say is that these targets are not equal. The black target is more vulnerable for whatever reason than the gray target. So that's what we became very interested in trying to understand is, are all essential genes equally essential in TB? And if not, can we find the most vulnerable of them, prioritize those as targets for target-based drug discovery, and then might we tip the scales in our favor and get this to work? So that was the idea. Next slide. Okay, and so effectively what we wanted to do is for every target in the TB genome, we wanted to ask how vulnerable is it? We wanted to inhibit it a little bit, and then a little bit more, and a little bit more, and a little bit more, and then ask where's that break point for every target? How much do I need to inhibit every target in TB in order to kill the bacterium, or at least prevent growth? And the way we did this is this Crispr interference system. So instead of CRISPR scissors, this is CRISPR interference. So it sits down on the target gene and blocks transcription. So it's a transcriptional interference or mRNA interference mechanism, or it knocks down production of the mRNA by RNA polymerase. And what was nice about the system that we're using, it's different than the standard pyogeny system that most labs are using. And I would encourage you to take a look at this manuscript if you're interested in the details. But we can tune with this system. So if you go to the next slide, this is a reporter gene where just by changing what part of the gene that we're targeting, the PAM sequence, we can get a 200-fold dynamic range of that target. So we can knock it down a lot, 200-fold, or we can knock it down really little bit, say 30%. And so what we reasoned is by using this tunability, we could dial up or down the magnitude of knockdown for every target in the TB genome and ask, at what point the bacteria die? How much knockdown for every target do I need to get? So next slide. This work was led by a really fantastic PhD student, Barbara Bosch, in the lab, and an equally fantastic computational scientist in the lab, Michael De Jesus, in close collaboration with our dear friend, Dirk Schnappinger, across the street at Weill Cornell. Next slide. So here's what they did. And here's where our dear friends at Twist come into play. So if we want to do every target at a time, we need this to be a highly parallel assay. So what we do is we synthesize sgRNAs, oligos, for every gene in the TB genome. And we tune them in the way that we make strong sgRNAs for a gene, medium sgRNAs that knock it down partially, and then weak that shouldn't knock it down a lot. This ends up being a lot of sgRNAs. It's about 100,000 different sgRNAs for the about 4,000 genes in the TB genome. In unpublished work with Twist, we've gone up to 1 million sgRNAs, and that's also worked quite well. But what Barbara did is she cloned this array of sgRNAs targeting every gene in the TB genome, cloned it into E. coli, put it into TB, and now every single bacterium has a single plasmid that has a single guide RNA targeting a single gene in the TB genome. So it's this pool of 100,000 different bacteria. Next slide. There's Twist. Yeah, they built. Okay. And then Barbara did this competitive growth experiment. So our system is small molecule inducible. In the absence of ATC, doesn't matter what it is, the system is off. In the presence of ATC, the system is on. So what she did is she passaged this library over time, over about 30 generations. So this takes about a month in TB. Harvested aliquots of the genomic DNA over time. And then we simply count sgRNAs as a proxy for bacterial fitness. That's how many, the number of those bacteria present in this population. Okay, next slide. And so then came the fun part, where Michael really shone, is figuring out how to analyze this, figuring out how to quantitatively define a vulnerable versus invulnerable gene. So a vulnerable gene is a gene in the red line there that has a very high maximum fitness cost associated with strong knockdown. So if you inhibit this thing, there's a strong fitness cost to the bacteria. We wanted to know how sensitive is it to partial knockdown? How much knockdown do you need to get to impose that fitness cost? So that's sort of that medium gray line there. And then 3, what is the phenotypic lag? That's how long between when we turn CRISPR interference on to when the bacteria start to get sick. So a vulnerable gene has a very high maximum fitness cost, it's very sensitive to partial knockdown, and it has a short lag. An invulnerable gene is just the opposite of all of those three things. So that's what Barbara and Michael set out to do, is for every gene in the genome, quantify these parameters to define the most vulnerable genes. Next slide. Okay, does the system work at all? So the first thing we wanted to do, this is a brand new approach in our bacterium, is it producing data that make any sense at all? So we know there were about 600 to 700 essential genes in TB based on an approach called transposon sequencing. And so we asked, well, when we knock down these genes with strong guides, do they deplete from the library? And so you can see this blob in this heat map here. Each row is a gene. You can see each column is a passage of the library, and you can see a blob of genes that depletes on the upper right-hand corner. So if we define genes as essential by CRISPR-I and compare that to TNC, the overlap is pretty good, right? Just benchmarking our data, the data make sense. Okay, so at that point, and I won't show you a lot of further confirmatory data, we got to do the fun stuff. So next slide. So we want to know how much knockdown are we getting for all 96,000 guides in the library. That many guides, you can't obviously go and do a Western blot for every single one or a qPCR, that's not possible. So we had to computationally infer how much knockdown that we're getting. So what Michael did is, these are 3 different guides targeting an essential gene in the TB genome, and red is strong, blue is weak. So you can see the red guide depletes quite rapidly from the library, and the blue guide doesn't. The middle one is somewhere in between. So he fit a 2-line model for all 96,700 guides in the library. And beta E is effectively the fitness cost imposed by that STR. It's just the slope of that line. And then he fed this all into a linear model, just a very simple form of machine learning, and asked what features of a guide predict how strong it's going to be. And that's the next slide. And the results were not surprising. They were what we would expect. The features that dictate the fitness cost imposed by a guide were what PAM was that sgRNA targeting. We have strong PAMs and weak PAMs. How long that sgRNA targeting sequence is, which is another parameter we use to tune, and then the GC content of the guides. So we went on to confirm that these parameters were validated with an independent set of sgRNAs, but this wasn't biologically interesting, but it gives us quantitative predictions for how strong every guide is in the library. We can just take a look at those parameters for every sgRNA and give it a score. 0 being weak, 1 being very strong. And color code them: red, strong, blue, weak. So if we go to the next slide. So these are actual data. So MMPL3 is an essential gene in TB, and you can see it depletes quite rapidly over time because there's a very strong fitness cost associated with that, knocking that gene down. Dark blue guides don't deplete, and we can get every quantitative phenotype in between because of the scale at which we can work. FLIGR, doesn't matter what it is, it's a non-essential gene. It doesn't deplete over time. Okay. So now we have these line plots for every gene. We have predictions for how strong every sgRNA is in the gene. And now what we want to know is, what's the gene level vulnerability? How much knockdown do I need to get for every gene in the TB genome to impose a fitness cost? So now we need to take this data and aggregate it into a gene level call. And so what Michael did, next slide, is estimate this expression fitness relationship so on the x-axis is the sgRNA predicted strength, which is our estimate of how much knockdown we're getting, right? It's a proxy for that, versus the estimated rate of depletion, which is the fitness cost associated with a guide of that strength. So Michael fits a hill curve for every gene that again estimates this sort of expression on the x-axis, fitness on the y-axis for every all 4,000 genes in the TB genome. Go to the next slide. This is what that actually looks like. So here are the actual data. This is Bayesian, so he iterates through it 10,000 different times. Each dot is an sgRNA, and you can see these hill curves in gray. Each gray line is a single logistic regression or hill curve from each individual iteration through this model, and then black is sort of the mean logistic regression curve. So you can see there's some noise in the data, but the fit looks pretty reasonable there. So he could do this for all 4,000 genes in the TB genome, and now we finally get to get some answer to our question: are all essential genes equally essential? So if you go to the next slide, and the answer, thankfully, is no. So These are 4 different essential genes in the TB genome. So if you delete any of them, the bacteria can't grow, but you can see they have very different expression fitness relationships, right? With the dark purple one, MMPL3, being very sensitive to inhibition, and in the sort of pink and light gray, gene MOB1, another essential gene, is highly invulnerable, right? It has a weak maximum fitness cost associated with strong knockdown, and you need to knock down a lot in order to impose that fitness cost. So essential genes have widely variable expression fitness relationships. They vary widely in their vulnerability. So these are 4 different essential genes. We can do this for all 4,000 genes in the TB genome. Go to the next slide. So here's what it looks like for every gene in the TB genome. Each dot is a gene. The closer that dot gets to the center of the bullseye, the more vulnerable that gene is. So there's sort of 2 take-home messages here. So in blue, the blue dots are the targets of the first-line therapy for this bacterial infection. And you can see they're vulnerable, which validates this approach. These are drugs that won out in the clinic. But in red are the upper quartile, the most vulnerable genes in the TB genome. And as you can see, there are genes that are much more vulnerable than sort of our best standard therapy than we have now. So that means there's hope that in principle we could do better than the drugs that we have now. The second take-home message is related to the previous slide, is that you can see that the vulnerability really varies quite widely across the TB genome. Again, not all essential genes are equally essential. Next slide. So in the last slide or 2, well, what are the most vulnerable genes in the TB genome? And I won't dwell on this. Some of them are exactly what you would have expected, right? Central dogma. You need to be able to replicate your chromosome. You have to turn DNA to RNA. You have to turn RNA into protein. But they weren't all what you would have expected. This protease called the click protease, for example, is essential and highly vulnerable in TB. We have absolutely no idea why, but we're hoping to figure it out. One of the most striking high-level overview or enriched classes of invulnerable genes were a lot of metabolic genes and genes involved in, for example, coenzyme A, biosynthesis, sulfur metabolism, that as a whole were exceedingly invulnerable. It's as if they are present at levels much higher than needed for maximum fitness in bacteria for, again, reasons that are interesting to speculate about. We can look at sort of the pathway level. We can also then zoom in and look at the individual pathways. So, DNA replication, for example, as a process is highly vulnerable, but within DNA replication, the main polymerase is exceedingly vulnerable, gyrase is exceedingly vulnerable, but Okazaki factor maturation, which is essential, is exceedingly invulnerable. So when you're thinking about drugging DNA replication, for example, the main replicative polymerase is solely viewed through the lens of vulnerability, a much more attractive target than enzymes involved in Okazaki factor maturation. Okay, so last slide, or second to last slide. As I mentioned, gene vulnerability varies widely across the TB genome. Not all essential genes are equally essential. Next slide. Bringing this back to why we started this. So what does this mean for drug discovery? If not all essential genes are equally essential, by this metric, not all drug targets are made equal. So here, RPOB is a subunit of RNA polymerase. It's very vulnerable. It's really a fantastic drug target in TB. DEP is a peptide deformylase involved in translation in microbacteria. It's essential. Companies and academic labs have spent a lot of time and resources trying to drug this thing. It's an exceedingly invulnerable target. So you're perhaps not setting yourself up for success by picking a target like this. So ignoring druggability, these sorts of questions, if we view this solely through the lens of vulnerability, which I don't entirely advocate for, but it's one important aspect of this, all else being equal, prioritize vulnerable targets over invulnerable targets for target-based drug discovery. Again, asterisk, all else being equal. Okay, next slide. So that's it. So vulnerability, what I hope I've convinced you is, yes, chemistry matters, compounds absolutely matter, but so do their targets. And with that, I just thank my lab, which I think is the acknowledgement. So I'll thank Barbara and Michael and the rest of the lab that's involved in these studies, and everyone here for their attention.
All right, are there any questions for Dr. Jennifer?
In the back.
Yeah, so for the vulnerable genes, are any of these concerned? Are you concerned about having like homology with human targets in terms of like side effects for targeting these things?
Yeah, that's a great question, Brennan. Yeah, so the question is, what if the bacterial target has a human homologue? Yeah, so sometimes that can be a deal breaker, right? If the two enzymes are highly homologous, it can be very difficult to develop an inhibitor that's specific for the bacterial homologue. Doesn't mean it's not possible. For example, Carl Nathan at Cornell has developed proteasome inhibitors that are specific for the TB proteasome that don't hit the human proteasome. But it is absolutely something that one would want to keep in mind when picking a target.
Since two of the main ways that the bacteria are evading small molecule inhibition is degradation and efflux pumps, do you think overall vulnerability or sort of the rate at which it is inhibited is more important, or do you need to balance the overall vulnerability with how quickly the bacteria is stopped from replicating?
Yeah, good question. So are you thinking, like how fast your inhibitor needs to act before it gets metabolized or before it gets kicked out by efflux pumps? Mm-hmm. Yeah, it's an interesting, I think it's a very good point. Yeah, and here's where something like if you could get, say a covalent inhibitor, right, that's going to latch onto that target and then it's not going to get pumped back out. Something like a covalent inhibitor might be able to get around these things. But yeah, absolutely. Once you get to actual compounds, then all of these parameters become very important and definitely something to keep in mind.
We have actually one question from online. How does your vulnerability metric fit into traditional druggability?
So another great question. So we don't take druggability into account at all. And for a few reasons. One, our approach doesn't address druggability at all. This is solely a genetic approach, quantifying this sort of expression-fitness relationship. Druggability is super important. If your enzyme doesn't have a druggable pocket, then, again, you might be facing an uphill battle. So we view this vulnerability approach as really working hand in hand with many other metrics that one might use to nominate target for target-based drug discovery, including things like druggability. That said, I would highlight that our definition of vulnerability continues to expand. Things like PROTACs, right? We don't need an active site, just any small molecule that can latch on there. So I would argue that, yes, you absolutely have to take it into account, but things that are not druggable now may be druggable in the future. Yeah, Brennan.
I have another question. So obviously, you've got a lot before you can run, so this may be a question about your stage of research, but have you tried to do any knockouts of multiple vulnerable genes to see how they sort of work together? Are you still in the classification stage of individual genes?
Another great question. So yeah, my PhD student Isaac is on the call. And Barbara, that is one of their big next steps is now we can knock down each gene individually, but now we can start to think about genetic interactions. Like how are these genes interacting with each other? Can we identify these sorts of, super synthetic lethalities between two vulnerable genes? Absolutely. And here's where being able to look at something like 1 million sgRNAs in pairwise combination, not 1 million squared, but not quite that many. But yeah, it becomes super important and is very much an active area of work in the lab.
I thank everyone for joining us online and here in person. If you will do us a big favor, there will be a quick 1-minute survey right after this wraps up. And if you could fill it out, that would really help us. So thank you all for attending.